Welcome to your 14th laboratory exercise, or the second part of the identification of the members of family Enterobacteriaceae. This will include the following tests: urease production, decarboxylation and deamination in lysine iron agar, the different IMVIC tests, and motility testing. First, let us have the materials for urease production. An inoculating loop, alcohol lamp, bacterial broth culture, usually of Proteus and Escherichia, Christensen's urea broth. Now for the procedure of urease production. First is to label your tubes properly. Using a sterile inoculating loop, get an inoculum from the bacterial broth culture. Inoculate the Christensen's urea broth with the loopful of the organism using the dip and shake method. Incubate at 37 degrees Celsius. Observe for color change within 2 to 4 hours. Generally, the reaction is completed within 24 hours. The indicator used in this test is phenol red. The positive result is the appearance of bright pink color usually seen in Proteus, while the negative result is the no change in color of the yellow medium usually seen in E. coli. Now we go to the materials for the reactions in LIA. First is the LIA, or the lysine iron agar, inoculating needle, alcohol lamp, and your bacterial broth culture of E. coli and Proteus. Now for the procedure for reactions in LIA. Using a sterile inoculating needle, get an inoculum from the bacterial broth culture. Inoculate the LIA using the inoculating needle via the stab and streak method. Stab the deep or the butt end and streak the slant. Incubate at 37 degrees Celsius for 18 to 24 hours. If necessary, incubate for 48 hours. These are the possible reactions in LIA. The tube on the rightmost is the uninoculated tube for comparison. Lysine decarboxylation, or LDC, occurs in the butt, while lysine deamination, or LDA, occurs in the slant. H2S production is seen as blackening of the medium, while the indicator used in LIA is bromcresol purple. In the first tube, if the organism produces lysine decarboxylase or it is LDC positive, cadaverine on the butt is formed in the form of a purple butt. If deamination does not occur or it is LDA negative, the slant remains purple. This is an example of a purple slant over a purple butt, meaning an alkaline slant over an alkaline butt, so this is reported as K over K. This is another example of an alkaline slant over an alkaline butt, or K over K, but is accompanied by a black precipitate, which indicates the production of hydrogen sulfide. So this is reported as K over K H2S positive. In this next tube, if the decarboxylase is not produced or it is LDC negative, the butt remains acidic or yellow. Now, this means that glucose was fermented, and when glucose is fermented, the butt of the medium becomes acidic or yellow. And again, if it is LDA negative, the slant remains purple. This is an example of a purple slant over a yellow butt, or an alkaline slant over an acidic butt. This is reported as K over A. This is another example showing LDC negative. The butt remains acidic or yellow and glucose was fermented. However, if oxidative deamination of lysine occurs or it is LDA positive, the color of the slant will form a red color in the presence of ferric ammonium citrate and flavine mononucleotide. This is an example of a red slant over a yellow butt, or a red slant over an acidic butt. This is reported as R over A. Next, we proceed to the materials for IMVIC test. We need the Indole or SIM or SIM medium, MRVP broth, Simon citrate medium, inoculating loop and needle, methyl red reagent, alpha naphthol reagent, and sodium hydroxide or NaOH. IMVIC consists of 4 tests, and the first one is indole production. Indole production is the letter I of IMVIC, and the principle of this test is it determines the ability of an organism to split tryptophan to form a compound known as indole. Using a sterile inoculating needle, get an inoculum from the bacterial broth culture. Inoculate the indole or SIM medium using the inoculating needle via the stab method. Replace the cap loosely and incubate at 37 degrees Celsius for 18 to 24 hours. After incubation, add 5 ml of COVAX reagent to the SIM. The positive result would be the formation of red colored ring after the addition of COVAX reagent. The next tests are the letters M and V of IMVIC, or the methyl red and Voges-Proskauer test. Methyl red and Voges-Proskauer, or MRVP, are 2 different tests. Methyl red detects the production of acid by an organism through the development of color change, while Voges-Proskauer detects the presence of acetoin. Using a sterile inoculating loop, get inoculum from the bacterial broth culture. First, inoculate the MR broth with the loopful of the organism using the dip and shake method. To repeat the same procedure, get inoculum again from the bacterial broth culture, and this time, inoculate the VP broth with the loopful of the organism using the dip and shake method. Incubate the MRVP tubes for 48 hours or until sufficient growth occurs in the broth. After 48 hours, take out the tubes for MR and VP. First is to perform the MR test by adding 5 drops of the MR or methyl red indicator. Interpret the color results immediately. The positive MR test is the appearance of a distinct red color at the surface of the medium. The appearance of an orange color is considered a delayed reaction. In this case, continue incubation and repeat the test in 4 days. No attempt should be made to interpret an MR test before 48 hours, since false positive results may occur. Perform the VP test by adding 12 drops of alpha-naphthol reagent and shake well. Next is to add 4 drops of the sodium hydroxide or NaOH reagent. Gently shake the tubes for 30 seconds to 1 minute to expose reaction to atmospheric oxygen. This oxidizes acetylene to obtain a color reaction. Allow the tubes to stand for at least 10 to 15 minutes. The positive result for VP is the formation of a pink to red color at the surface of the medium. And the last of the IMVIC test is the letter C, or the citrate utilization test. The citrate utilization test determines if the organism utilizes citrate as its sole source of carbon. Using a sterile inoculating loop, get inoculum from the bacterial broth culture. Inoculate the citrate slant using the inoculating loop by simple streaking. Leave on the cap loosely and incubate at 37 degrees Celsius for 18 to 24 hours. The positive result is indicated by the change in color from green to blue in the citrate medium. And lastly, we have the motility test. The materials needed are the SIM or SIM medium, inoculating needle, and alcohol lamp. For the procedure of motility testing, using a sterile inoculating needle, get an inoculum from the bacterial broth culture. Inoculate the indole or SIM medium using the inoculating needle via the stab method. Stab the needle up to the bottom area of the agar. Incubate at 37 degrees Celsius for 18 to 24 hours. Examine for growth around the line of inoculation. The bacteria is considered motile if there is diffuse growth extending laterally from the line of inoculation seen in the left side of the picture, while the bacteria is non-motile if there is growth only along the line of inoculation, like what is seen in the right side of the photo.